Hi, I'm Priyal D'Souza. Welcome to my talk. I'm going to talk about clinical genomics, um, the basics and an up-to-date technological perspective of clinical genomics, in fact. What is clinical genomics? Well, clinical genomics is a bottom-up approach that uses DNA sequences to identify changes resulting in molecular mechanisms which are likely to cause disease. It has become a more frequent way of diagnosing many inherited diseases, as well as cancer, and it's the present way of doing it, and it definitely will be in the future as well. On this slide, I'm going to talk about common types of genomic analysis which uses DNA as a starting material. One of the first analytical procedures was a single gene amplification followed by sequencing and mutation or variation detection. That particular analytical procedure was started in the late 80s and has survived for quite a few decades after that. The turn of the century saw the beginning of microarray based DNA hybridization panels, which was largely used in R&D settings to try and work out diagnostic tests which were basically went back to single gene amplification. And in the last decade, next generation sequencing has come on board, and this enabled parallel massively parallel sequencing of simultaneous chunks of DNA and enabled the whole genome to be sequenced in a very short period of time. Is days opposed to months and years. The whole exome is also the coding parts of the sequence can also be done with next generation sequencing, as well as these specific techniques called chip or chromatin immunoprecipitation sequencing or methylome sequencing. All these techniques are really used by the R&D labs to develop diagnostics, so they're not readily used in the clinical settings. The technique I'm going to talk about is target-enriched next-generation sequencing panels, and that is actually used quite a lot in the clinical diagnostic settings. It can also be used in the R&D settings, and it's been around since about 2013. So targeted enriched next-generation sequencing panels, they're a commercially available solution, an end-to-end diagnostic solution that you can buy from biotechnology companies. And these panels tend to be disease specific, as you'll see in the following slides. The assays that are available when you use these panels have been validated, and they meet regulatory requirements in clinical environment. So what is an NGS panel? NGS panel consists of several regions which could be adjoining each other on the same gene or on the same chromosome or on different chromosomes. So it's just showing here regions of one gene. And if you look at it in more detail, what this region consists of are sequences where there are to be known mutations from previous established studies are called mutation hotspots, and these hotspots are likely to cause disease. And this panel consists of thousands and thousands of these mutational hotspots right across the genome, and these particular amplification methods are chosen so that all simultaneous massive parallel amplification be carried out in a single test tube. In the worst case scenario, it can be carried out in two different test tubes. So, going from a single gene in one tube to, thousands of different loci in one tube is a massive jump, and speeded up the whole process of clinical diagnostics. So the next slide So I'm going to talk about some examples of NGS panels which are out available commercially. This is only a very small section of the actual panels available. Typical panel will contain anything between 15 to 50 different genes, and the four panels here are a bladder cancer panel, a kidney carcinoma panel, a BRCA or breast cancer panel, and it's a liver panel here in the bottom right hand corner. So how do we use these panels? Well, you need to make DNA from a blood or a tumor sample and follow this workflow here, which basically you start by making a DNA library, and you prepare a template, and you run the sequence, load the template onto a sequencing machine, and then that automatically generates data which get processed, and a final report is made which can be accessed by the pathologist or the clinician. These particular DNA preparation steps are normally carried out by scientists rather than clinicians, and once the data is obtained at the end, then basically we can actually start interpreting the data, and that's normally done by the clinician would ask the pathologist to review the automated report and suggest a diagnosis according to the mutation data that's produced. That's it. Thank you.